Ekranı 5400'lü alırsan eğer işte havaları uçuyoruz. Evet. Hiçbir şey çok düşmüşsün. Ama şey var. Ya
Okay. Then let's continue with the biological basis of EEG, or let's call it maybe also the standard model of EEG, the standard biological model of EEG. And we now zoomed into the cortex, into this pyramidal neurons, which are having their cell bodies in layer 5, but extend quite a long way with the apical dendrites up to the surface of the cortex, and of course extend their axons either to other cortical sites, or if you're in the motor cortex, of course also to the periphery. What this shows here is this typical picture of a neuron that you are very familiar with, and you know all the parts here. This taken from Niedermeyer's EEG book, maybe shows a little bit more realistic in the sense that there are everywhere on the neuron there are these dendrites coming from elsewhere, and of course there's also glia somewhere. So the whole thing is of course it's not sitting there isolated in the pyramidal neuron, but there are many dendrites that are connected to its sorry, am I saying the right word? Postsynaptic sites, that are connected to the dendrites. So this is the end of the axon of another neuron, which is called the presynaptic site. And they are making synapses here at the dendrites of the neuron. And then you also know this very well, that this shows a typical experiment that you can do in kind of a petri dish to induce the phenomena that you are familiar with from undergraduate neuropsychology, maybe, that you can induce two sorts of things depending on the synapse, namely excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And this here shows a experimental setup. So here you have an electrode that's measuring the, maybe I should use the pointer, sorry. Here you have the an electrode that measures the presynaptic activity of a dendrite that forms an excitatory synapse with the pyramidal cell neuron. So here you have some spiking activity going on, and then you can see that there is a little bit of a build-up of an excitatory postsynaptic potential at the postsynaptic neuron, the pyramidal cell neuron that we're interested in, which is measured here. And if there's enough excitatory presynaptic activity, so in this case 2 spikes here, then this excitatory postsynaptic potential might be large enough to be supra-threshold and induce an action potential. That's a classical story that you all know. But what's also happening, of course, and can happen, is that you have inhibitory presynaptic activity, which induces inhibitory postsynaptic potentials, so making potentials more negative here at the inside of the neuron, for example, by the opening of chloride channels. So this is the classical thing that you should definitely know from undergraduate neuroscience, let's say. One thing that is important for EEG is not so much the intracellular activity, but also that the intracellular activity links with extracellular currents. So if something is happening and you are familiar with on the inside of the cell, there's also something happening outside. So this shows an example of an excitatory potential, where of course what's happening is that because you have positive ions flowing in, you have extracellular movement of positive charge towards the side of the synapse. So you have this extracellular flow that actually basically goes in this direction to here and goes from here to here in terms of positive charges outside. And of course, the reverse happens if you have negative, also inhibitory potentials. Then the positive ions flow away from the presynaptic terminal in the outside medium. What else should happen? There's no surprise here. Now, these kind of extracellular current flows and their associated potentials, they are what is measured with the EEG, but also with various other electrical or electrophysiological measurement techniques. We will see a little bit more specifically what the EEG picks up, but in general you should be aware that these different techniques, they are all sensitive to essentially the same kind of biological currents. So these kind of extracellular activities are called field potentials, and specifically local field potentials, if you put an electrode into the brain outside of the neurons and look at what kind of electrical activity you pick up for this electrode with respect to a reference electrode, of course, and you look at the potential evolution. So that's what is called local field potentials. These are potentials, and local field potentials, as I mentioned earlier, doesn't really make sense. So these are just potentials. So they are not electric fields, but they are electrical potentials that are picked up by these electrodes. And you can measure them there, and for maybe still they are considered like the gold standard maybe of invasive or of cognitive neuroscience. But you can, of course, because that's very close to the neurons, but you can pick up the same kind of activity but filtered through things that, of course, happen between this side where this electrode sticks and another electrode at the cortical surface. So I will show you a picture in a second. Of course, you can, and sometimes have to, in patients or in animal subjects, put electrodes directly onto the cortex, so not onto the scalp, but onto the cortex. Then it's called electrocorticogram, and they measure also this the potential varying over time, which is happening in the extracellular space. And of course, the EEG that you put onto the scalp picks up the same kind of activity. But, and this is of course the important thing, is that they filter this kind of activity in different ways. But the origin story for all the signals that can be observed in either invasive electrophysiology in monkeys with the local field potentials, sometimes in humans with the electrocorticogram on the cortex and the EEG, the basis for that is always this extracellular flow of ions. And of course, the MEG measures the associated magnetic field. So also MEG, like we discussed last time at some length, picks up essentially the same signals. Now, the thing that then gives each of these characteristics somehow their specific ways, of course, where the electrode is placed, and then of course also how whether you pick up the magnetic field or the electrical activity. But there's one source that gives rise to these things. Yeah.
So the difference between the COG and the electrode is just the size of the electrode?
The side of the electrode, where it is. where it is, so where it's stuck. Yeah. So it's not that this is something different. So this is actually, maybe I go back to this picture in a second, but this is, so this kind of thing, this concept that I'm telling you about, this is based on this Bujaki 2012 Nature Reviews Neuroscience review, which is very nice and which makes this point very, very clear. So this shows an example, although this is not in complete spatial alignment here, but it makes the point that there are a lot of electrical things going on in the extracellular space, and we will look at them in more detail for the EEG in a second, but there's a lot of stuff going on with these EPSPs and IPSPs. They get summed over space, and if they somehow add up, according to the superposition principle, they might be detectable. And now LFPs, ECOG and EEG and MEG are sensitive to these kind of activities, but at different distances from their origin. Yeah? And this is what is essentially shown here, that so if you're closer with some depth electrodes, so this is from one patient who had depth electrodes, but also ECOG electrodes and EEG. What you typically observe is stronger or a little bit more higher amplitude variations when you are invasive, but kind of qualitatively the data are fairly similar. So there is some work by also Nikos Logothetis, and the first author, which I now forget, where they recorded monkey invasive physiology and EEG activity, much as they did in this paper for the fMRI, and then tried to figure out where the highest correlation is, in which frequency band. There's a second, maybe I show that later, or maybe I showed it in the other class, where they then tried to find correlations between different frequency bands of the invasive data and the EEG. But this is, one can do that. The thing is, and this is also something that you start to realize, is that you always have these measure sensors and you can put them somewhere and then record some data, but that doesn't mean then that the data explains to you how the brain works. Then you need models and data analytical things and correlations between data analysis to make some sense of it. Essentially, you need your own creativity to make some sense of it. And the thing is, it's not that the LFP electrode will tell you the truth, so what's going on, and the EEG electrode will tell you not the truth of what's not going on. They detect the same overall activity, but at different distances. So I really recommend reading this review by Bujaki, because it's kind of up to date. It doesn't feel like it's written in the 70s and today we can do all these more fancy things. No, it's from these days, 6 years now. And here are just pictures of, to give you a feeling of how these things are measured that we just discussed. So the local field potentials with, in this case, with depth electrodes, as you saw also in the talk last week in CCNB, in monkeys, in the good labs actually, which are also now closed down, of course. This is, I think, from Nikos Logothetis lab, where they don't do this anymore, where you have these chambers where you can put in the electrodes, and everything is fairly clean. So it's not that the monkeys run around with their whole or half of their skull removed, but it's like this, like in patients. Electrocardiogram here in monkey with a little bit more stuff removed. So we had a talk on this by the guy from Frankfurt, what's his name? Pascal Fries. So he has been talking about the electrocardiogram work they did in monkeys for some time now. So they had these grids on monkey brain, which this I think is also monkey. I would say, from the anatomy of the skull, where you can see this grid of that's always called subdural electrodes. So basically, you put EEG electrodes. Essentially, that's what it is. You put EEG electrodes not on the skull, but on the cortex after removing the skull. That's how that looks. And of course, we've seen MEG. So,
isn't that really painful?
What is painful?
Putting the electrodes through the head like this.
This?
No, on the left.
This? Or this? The monkey?
Yeah.
I mean, the brain doesn't have pain
I
sensors.
know, but your head.
Yeah, of course. So, of course, that's painful. So this is why this is implanted under anesthesia, obviously. But especially for these, if they do that in patients with the depth electrodes, I think they also they mainly do local anesthesia to also then test immediately, like in these old stories that you read. But of course, yeah, I mean, in both cases, in the animal experiments, in the humans, of course, it is ensured that when the surgery happens, that the subjects don't experience pain. I mean, you couldn't. I mean, with the monkey experiments, you couldn't do any experiments on monkeys that experience pain. So they will not. I mean, these monkeys have to do experiments that are really boring. So they have to look at. Look at some fixation dot, and then sometimes they get a little bit of juice if they look to the right point at the right time. If they would be under pain, they would not do anything at all. So, of course, in any case, in animal experiments and also in human not human experiments, in this case in human treatments, of course, it's ensured that there's no pain associated with that.
And for the electrocardiogram, do you have to remove half of? So afterwards, they are able to live? Is that the end of it, or can they live on after?
Well, I mean, the monkey experiments, they are in the end, they are always terminal, of course, because in the end the monkey will die. But of course they get implanted, and usually, and of course the skull. So they also don't run around like this, but they put the skull back on. And for human participants or human patients, they use this ECOG data also to put stuff on the temporal cortex to figure out what's going on with epilepsy. And of course, they close the skull again. And humans, then of course, these are not terminal experiments in humans, of course not. It's not crazy. Yeah, of course, then they can be removed again.
Why are they terminal in the case
yeah, that's a good point. So definitely in terms of these experiments, they are definitely terminal, because there you want to know where the electrode was sitting in the very end. And in these, I said they were terminal. Maybe you also want to you want to do the anatomy in the end. Maybe they are not terminal, I think, but you definitely, these monkeys, they will not be brought back to Africa or wherever they're coming from. I don't know where monkeys come from. And will be, I mean, animals used in human research, in the end, they usually die. But as you are also aware of that, which is also, of course, a good thing, that monkey experiments are under high pressure, especially in Germany, and it's getting harder and harder to get the ethics approval, which is also a good thing. So I'm not a big fan of monkey experiments, so don't believe that this adds too much to the whole cognitive neuroscience thing. Yeah. Good, and of course we also don't want to do these kind of experiments ourselves. And I'm also not a person who believes, a lot of people believe that also in cognitive neuroimaging, that we can only make good cognitive neuroimaging if we finally know what our signals mean. I don't believe that, because you also don't know what these signals mean that you get from the invasive recordings. So you have to do all sorts of filtering, data analysis, and so on to have some interpretation of what's going on. And I think it's much more important that that this thing of like, but the neurons with the action potentials and the postsynaptic potentials, Where are they in neural imaging? I think it's much more important to put them into data analytical models for EEG data, like we do, so that you have biophysical modeling, so that you feel happy and satisfied from a neuroscientific perspective and acknowledge that you would have to do the same thing if you would stick an electrode into some monkey's brain, because the electrode in the monkey's brain doesn't tell you the truth, it just gives you a curve. So, but some people believe that you need to validate these models based on invasive data, and I don't think so. You need to validate the data, the models, of course, but with respect to other models. I need a model comparison, but this is a longer discussion. Good. So that was kind of the general thing that's happening, and now we want to, but there's a little bit more to the EEG, because it's not like this is just the extracellular stuff. One can say a little bit more for EEG in terms of the standard model. And this story goes as follows. Is there a question? Or, no? This story goes as follows. If one has one thinks essentially, and we can get back to that slide in a second, one thinks about these pyramidal cell neurons that we have seen in the Golgi stain, and thinks about what's actually happening if they get input and get activity input at their synapses, which are located at their dendrites. Maybe I explain this picture first, or also only because the other is not helping that much. So the idea is as follows. We've seen that at the site of the synapse, what happens is that there is a flow into the neuron, which induces an extracellular flow, an extracellular current. And the important thing is that if you have some current at one point that flows somewhere into the cell, which is extended in space like this, these charges, well, they must come from somewhere. And the general idea is that if you have some current sink somewhere where charge flows into the cell, then at some other place this charge must come from, and in this case it's coming from a current source that is is the same cell. And the most important thing is that you get an extracellular current that extends over this extent of the pyramidal cell. So let's go back to the other slide here, which makes this a little bit more fine-grained, if you want. First, there is some rest, and you have some synapse here. The intracellular medium is charged a little bit more negative than the outside. You know that the resting state membrane potential is something minus 60 millivolt or something, so it's negative inside. And then the synapse is activated, and cations, so positive ions, flow through the membrane. Yeah. So now you have movement of positive neurons in there, which induces along the membrane in the outside medium. So this little thing, do you see this? Not. This little thing here. Do you see the hand? Maybe I should check whether you see the hand. You see this? No. Oh, maybe like this. One can only point in the small resolution. Sorry. this thing that's measuring the outside potential, because there is some flow here, this thing gets a little bit negative compared to this, which means that there is a potential difference in the outside, and then this third step happens. So currents along the membrane in the extra and intracellular space happen to make this thing happen. So somehow there is movement towards, in the outside, towards this side, and there's movement of corresponding charge inside. It doesn't necessarily mean that here you have to have an open synapse, not at all. It just means that you have current that basically tries to undo the change in potential here, and you have inside current that then tries to undo the local change here. So it doesn't need to be that here you have an open synapse as well, right? So it just flows something here, and to basically to make up for that at this point, you also have current flow. So yeah.
Can you say that again, zoomed out, so we can see what you're talking about?
It doesn't show what you're indicating when you're zoomed in.
Ah, sorry.
Yeah,
like I realized. So the story is you shouldn't. So you shouldn't think that it's more complex than you would think intuitively. We are not in the other course. In the other course, everything is always more complex. Here, things are always more easy than they look. So here you have a synapse. Positive stuff is flowing in from somewhere else. Positive stuff is flowing here, and because then also here something changes, there's also some movement of charge in here, not necessarily through the membrane, but just to undo the change in potential here. Yeah. So the story is really, really simple. The story is there's a synapse. You all know that in a synapse, current is flowing into the cell. Well, from somewhere this current must come. And then the story is, well, it comes, in this case, so there is some current induced in the extracellular medium, and this current then extends over space. And this, if now, and this is the important thing also on this and also then on the next slide, if this is happening in parallel in a lot of these pyramidal cells, then then you have the setup of a current sink and a current source. So you essentially have an electrical circuit with a negative pole and a positive pole for some time. Yeah?
Why the current is from positive to negative is because the concentration of positive ions are larger?
Yeah, don't pin me on the. So you mean why here you have some?
Yeah, why negative? Yeah, why the direction is from positive to negative?
The main thing is that you here. So first you have this. Here you first have a resting balance state. So everything is balanced. And then something happens, namely that some positive ions go in. And they go in, of course, because there's some negative stuff. And then the story is just that you have a sink of charges. So at this point, the charges disappear. So to make up for this thing that charges disappear, there's some Ausgleichsstrom. So there's some movement of charges to make up for the missing thing. So to come back to the resting setting. So essentially, you can also view it differently. You can view it like you have a line cord here. Then you cut it and let electrodes. So let's say there's nothing running here, but there's the potential to run. And then you cut it, and then electrons are leaking out. And then because it's full of electrodes, they start to leak out there. Then the other electrons also start running, like that. Yeah, of course you would not. Maybe you wouldn't be quite happy from a physics perspective, because here no electrons will leak out. But nevertheless, that's the story. So the main point of this whole thing is just that. Opening of synapses induces currents across the membrane, and these currents across the membranes induce currents in the extracellular space, which I think is fairly okay. I mean, if there's some current somewhere, then of course the movement of charge must happen to that place. Now, before maybe not. What this induces, and this will then come, especially if you do source localization, the typical setup that this kind of, or the typical physical description of such a setup where you have a negative side and a positive side is then the electrical dipole, which of course you can then talk much more about in physics. But the fundamental idea is that if you have an axon, if you have a dendrite which has some inputs from other axons at synapses, this induces a current sink, a current source, and physically from going away from it, this looks like a thing that has a positive and a negative pole, because there is some delocation of charge. And these dipoles, they then induce themselves electric fields, hence electric potentials that you can measure far away on the EEG electrode. The whole thing is not that simple that you can say, okay, I see a deflection in the EEG like this. That means now these neurons have to be active. And this is what this next slide and the figure from Kandel I want to show. So you should not read into ERP polarity. This was also why I was a little bit worried about this question, whether it goes from plus to minus or minus to plus and so on. Don't worry about this. So the ERP polarity, so whether you have positive potential or negative potential, that depends on many things. Namely whether you have positive, in this basic model, whether you have positive or negative postsynaptic potentials, so excitatory and inhibitory, but also where that happens, and how this dipole is then actually lying in space, and how the recording electrode is in respect to that, and how the reference electrode is with respect to that. So the basic model of EEG is that you need synchronized activity in a lot of cells, in a lot of pyramidal cells in the cortical sheet. But if you, and this is what this figure shows, if you measure, for example, here now close to the electrode, because there's now a surplus of a positive charge, because here it gets more negative because positive cations run into the cells. Yeah, that can be the case, but this model is not identifiable, as I would call it, because the same thing can happen if you have well, this is not showing the same thing. Let's have another thing. No. The same thing could happen if you have if you have positivity induced up here, and vice versa. Jesus, this is so simple. And the thing is, either something is happening down here and it's negative, or something is happening up here and it's positive, and then you can do the 2x2 combination of that, and you get the same thing. Yeah. I don't. Obviously, I'm also exhausted at this point, so I cannot explain basic things anymore. But the bottom line of this is that you should not get too concerned with polarity and all these things. And you should also not believe in the standard model too much, and but you should basically have an idea of what people think, what they measure. And then if you really want to know, if you really think, this cannot be, I mean, what is this? So where's positive, where's negative? This needs to be clear. Then you start to do science. Yeah? So the basic, the bottom line is you have this input to the pyramidal cell apical dendrites, which are in parallel. These induce electrical currents here, and you can pick up the effects of these dipolar activity at the EEG electrode. Now, it is important that this then restrains of what you can actually pick up. So the good thing about the pyramidal cell neurons, as you've seen from the histology, is that they are arranged in parallel, and that what is called this open field configuration. So the idea is that if this is happening to at a lot of for a lot of cells at the same time, then just because of the spatial arrangement, these potentials that are induced, they don't cancel out as they do, for example, for subcortical nuclei, where the neurons and the dendrites are oriented inward or outward. So in this case, you could measure something if you have the electrode in here, but the contributions, if this is happening in time or parallel, of the neurons that are in there, they cancel each other out, and you don't detect it on the cortex. So again, now this is from the Luck book, and I think it summarizes really nicely the whole thing that I tried to talk about in the last 10 minutes or so in 3 figures. So the first thing is and is there something? Error options visible. No, still doesn't work. The first thing is that you have the pyramidal cell neurons, that these have dendrites, apical dendrites, and at these apical dendrites there are synapses, and then what can happen, and shown here for an excitatory postsynaptic potential, is that the outside of the outside medium accumulates negative charge. So at other places there's an accumulation of positive charge. So you get this dipolar setup, and this picture here then really nicely shows how you should think now going to from one to more neurons. So here you see these little things in the cortex. These should be now pyramidal cell neurons, and if this is happening synchronized in time, then there will be enough build up and enough strength of the potential that you might detect that from further away. So here you see that this whole thing has now on the outside something negative and on the inside something positive. And if you then zoom out further, you are in this case where where you see some negativity on the skull here. So the gray line here is now the skull, where you see some negativity here corresponding to some positivity elsewhere on the skull. The whole thing just induced by the local thing that is here called equivalent current dipole, but extending its electrical field and hence its potential through the whole of the skull, inducing what is called volume conduction, so that you see this electrical dipole orientation over the whole skull. And the simple model is that this, which we will now look at in EEG, where you always see some positivity somewhere, some negativity somewhere at specific points in time. You should, of course, always keep in mind that this is a dynamic thing, is based on the fact that the per millicell neurons are spatially parallel and aligned, and that they at the same time get a lot of similar EPSP activity. So let's see. So this is, of course, a simplified conceptual explanation of neural EEG origins. One important point is that what you pick up in this electrical activity, this is the summed thing of everything electrical that's happening. So there might be also fluctuations happening for action potentials, but they are not typically temporally aligned that much so that you'll be able to really see them. So in this, if you read this review, there are more things that contribute, of course, to EEG and specifically the extracellular potentials. Things like, to a certain degree, maybe action potentials, dendritic calcium spikes, maybe even, or not maybe, but also direct neural communication via gap junctions. And so on. So this is kind of, there's the textbook simple model, which, if you actually then look into the literature, is the working model for also EEG modeling. If you don't like that, I think that's too simple, then come up with better EEG models. It's a domain where you can do science, but what seems to be fairly certain are the following things. So if you want to measure something on the skull, then you of course need a large number of neurons. There are some papers that discuss how many exactly and how much of a cortical patch, but of course it's not if a single neuron is active and all the other neurons do something else. This does not sum up so that you can see it and will be filtered out. So it needs to be many neurons. They need to have the same spatial orientation, as we've seen from these closed field configurations for brainstem nuclei, which just physically cancel each other out. And the same thing then essentially saying that the neural dipoles must have a similar orientation. Yeah? So it's fairly simple. I would really say this basic model for EEG and what we see with the EEG is really simple. The idea is it rests on synaptic activity that gets into pyramidal dendrites. If this is happening in time, synchronized and synchronized in parallel over space, then we might see something. And of course, for the most basic event-related potentials, which we will talk about then from next time on, these event-related potentials, they are responses to but to very basic sensory stimuli, and they basically show the response of the brain to a very basic sensory stimulus, and where you can very easily think, okay, if there's a lot of light coming into the retina, then all the rods and cones and this whole business up to visual cortex induces some activity. And then, because the light is happening, this happens very fast, then at the same time, visual cortex, so the primary neurons in visual cortex, they all get this synaptic input. And then I kind of believe that if all these pyramidal cell neurons now at the same time get this, because you have the synchronized stimulus, that then there's this buildup of charge for a brief point in time, which you then can see on the EEG. So of course, I mean, for these sensory stuff, that's, I think, fairly convincing. For everything else, then it gets more tricky in terms of higher cognition and so on. But for this primary sensory stuff, I think this is, it's when you can measure if you have a neuron preparation in your petri dish and you give it a spike, you can measure some potential somewhere. So why should it be so different if you have it in vivo up to primary sensory cortex? So I don't think there's much magic there. Good. So that is the basic model for EEG responses, which you should learn about. And then next time we start to talk about event potentials. Any questions? No. Good. Then see you next Monday morning for active participation.